So I don't get an introduction because I'm just stepping in for somebody. So I'm very sorry if my talk lowers the tone a little bit, okay? So my talk has some clinical information in it because I thought we all like clinical information. And it's also got some quite old-fashioned but interesting functional work that generally doesn't attract the crowds, but it just shows what you can do with a very rare disease, a little bit of money, and some time on your hands. And I think it's kind of just something that we can think about for all the very rare patients that we get. So I'll have the next slide, please. And it's important just in terms of my association. So I'm now in France, employed by the French government, and I'm working at Cargo. And before I was at Manchester and Liverpool universities, and this work predominantly in Manchester. So here we have a family, and I apologize if the image is a little bit distressing. This is not a normal night out in Liverpool. This is a lady who had a child's shoe thrown in her face, and she suffered an expulsive hemorrhage and then had to have an evisceration. As you can see from her family tree, there are multiple consanguineous loops in her family, and you can see that there are four affected sisters who lost an eye through minor trauma. There's also some keratoconus in the family and some individuals with a blue sclera. This individual, individual 4-6, the other eye, as you can see, there's some extreme corneal thinning at a measure of 220 micrometers as opposed to the control 550. Next slide, please. So as I was saying, this family has a family history of ocular trauma and multiple adversities. So that means that that lady, unfortunately, had all her children removed and taken into care, and I think never got her children back because of alleged non-accidental injury. So if we move forward to the next slide. This is another family that presented with a less dramatic phenotype, which is why I think it's very important to talk about this family. The children had blue sclera, thin corneas detected on pachymetry, some keratoconus, deafness, and some skeletal features, including joint hypermobility. Next slide, please. So blue sclera, myopia, keratoconus, astigmatism, and thin corneas. So 275 micrometers in the affected eye. Next slide. So extraocular features included deafness, and I know there are some people in the audience who are interested in deafness. There's a specific feature in this family of hypercompliant tympanic membranes, bilateral moderate hearing loss, and also some progressive hearing loss, cleft lip and palate, developmental dysplasia of the hip, and joint hypermobility. Next slide, please. So in the laboratory findings in these two families, they had normal lysyl pyridinoline, hydroxyl lysyl pyridinoline ratios. So that differentiates this syndrome from EDS type 6. Next slide, please. So these are two cases of brittle cornea syndrome, which is a rare autosomal recessive condition characterized by extreme corneal thinning, corneal fragility with trauma, with rupture after minor trauma or spontaneously, and extraocular manifestations, which overlap with many features of connective tissue diseases. Next slide. In terms of the audiology, there's sensorineural hearing loss described and this feature of hypercompliant tympanic membranes. Brittle cornea syndrome has been associated with mutations in two genes, the gene ZNF469, and we identified mutations in this gene PRDM5 in these two families that I have just described from Manchester. Both ZNF469 and PRDM5 are transcription factors that are very poorly described in terms of what their function is, To date, ZNF469 is probably the more important of the two genes for various other reasons, particularly its association with corneal thinning in genome-wide association studies and its association with keratoconus in genome-wide association studies and candidate gene studies. But because it's such a large gene, it's difficult to clone and functional work has been very limited. PRDM5 and ZNF469 are believed to act on a pathway regulating extracellular matrix genes. Next slide, please. So PRDM5 is a very interesting gene. It's a gene of the family of PR domain containing proteins, and it's an epigenetic modifier gene that exerts sort of an indirect methyltransferase activity. So in terms of the mutations described in PRDM5, there's a cluster of mutations in the PR set domain, which is the protein-protein interaction domain, this in-frame deletion of exons 9 to 14, which was present in our family 1, and this stop mutation present in family 2. Next slide, please. So PRDM5 has been associated with repressive functions. Next, please, can you press? And the really interesting thing about a gene like PRDM5 and a disease like Brittle cornea syndrome, when you can get some material from patients, is that it provides an in vivo opportunity to observe a subset of epigenetic regulatory mechanisms in an inherited eye disease. So the interaction partners of PRDM5 were not well described at all. It was presumed that there was an interaction with gene INA, which is also euchromatin histone methyltransferase 2 and histone deacetylase 1. And PRDM5 loss of function had been described in cervical cancer. Next slide. So as I was saying, they're both implicated in the transcriptional activation of collagen genes, and with immunofluorescence, we can observe in both PRDM5 and ZNF469 mutant patient fibroblasts a decrease in the expression of collagens, fibronectin, and some integrins. Next slide. It's also interesting that obviously what you can observe in the skin fibroblasts may also be happening in the eye. And here is some immunohistochemistry, which shows the retinal pigment epithelium and Bruch's membrane and the choroid. And you can see in Bruch's membrane in the affected patients with the deletion of PRDM5 exons 9 to 14, that there is a decrease in the expression of type 1 collagen, type 4 collagen, and type 3 collagen in Bruch's membrane. Next slide. Which could be contributing to this patient with brittle cornea syndrome and a null mutation developing choroidal neovascularization at the age of 27. Next slide. Another thing that we observed was that PRDM5 was expressed in the cornea and in the retina, and that there appears to be a reduction in the number of retinal capillaries with a reduction of staining for collagen 5, collagen 1, collagen 3 in the small capillaries in the patients carrying the deletion in PRDM5. Next slide. So doing a general expression microarray on patient fibroblasts with mutations in ZNF469 or PRDM5, we could see all the genes that are dysregulated, and obviously the main thing that was noted was this general decrease in extracellular matrix genes. However, if you combine the expression microarray data with PRDM5 chip sequencing data and you only focus on those genes that are specifically bound by PRDM5, you can note an enrichment for PRDM5 target extracellular matrix genes with roles in vascular biology, and that these genes in particular appear to be dysregulated. Next slide. With an upregulation of extracellular matrix genes with roles in vascular biology like collagen 13, netrin 1, collagen 15, cadherin 5, with an enrichment for the repressive mark H3K9 dimethylation. Next slide. So, because we know that PRDM5 is supposed to be this epigenetic modifier, and we all know that our friends are important, we want to know PRDM5 interacting proteins and whether we can associate some of these retinal vascular morphological changes with disruption in PRDM5 function caused by transcription of some of these components and perhaps a loss of repressive activity of PRDM5 caused by these mutations. Next slide. So this is just basic biology, really. We did a co-immunoprecipitation combined with mass spectroscopy to confirm PRDM5's interaction partners. And we found that PRDM5 interacted with NERD complex proteins, so that's the very big, well-described chromatin remodeling complex, and proteins of the heterochromatin protein 1 family, the HP1 family, which is a large family of proteins which is believed to maintain the stability of heterochromatin. Next slide. In silico modeling suggested that the two missense mutations were predicted to interfere with the binding of the methyl donor S-adenosylmethionine and its methylation reaction product, S-adenosyl L-homocysteine. Next slide. So when we did mass spectrometry, co-immunoprecipitation combined with mass spectrometry with wild type and mutant constructs that contained this missense mutation within the PR domain, we noticed that in the wild type versus the mutant, there was a significant reduction in PRDM5's ability to associate with NERD component complexes, topoisomerase 1 and CHD4, which is the main enzyme of the NERD complex, and heterochromatin protein 1, HP1BP3, which is this heterochromatin protein 1 family protein. Next slide, please. We also demonstrated in the retinas of the patients and versus a control that the binding of HP1BP3 was very markedly reduced in the presence of a PRDM5 disease-causing mutation. Next slide. So in summary, clinically, there's now a diagnostic and carrier testing possible for brittle cornea syndrome. And it's very important that prevention of ocular rupture is very important. And these patients need to wear special polyacrylamide reinforced spectacles. I think the implications for the non-accidental injury scenario for all our patients with rare diseases is very important. In the UK, probably more than France, it's very common for patients and families with rare diseases to be involved in various social services interventions. So it's very important if we have a suspicion that something may be familial and we see something in accident and emergency and there's a strong family history, to actually document if we see any collagen-related systemic issues which were clearly present in these girls who presented to accident and emergency. And also be alert for combined sensory loss, as these children have both a potentially blinding condition and deafness. So when should brittle cornea syndrome be considered in differential diagnosis? When you have a thin cornea, a very thin cornea, with or without rupture, distinctive auditory findings like hypercompliant tympanic membrane and progressive deafness, which can be sensorineural, and a combination of musculoskeletal features like scoliosis, hip dysplasia, hypermobility. Next slide, please. From the research perspective, keep going, thank you. I think this work does provide some perspectives on the epigenetic consequences of mutations in PRDM5, and it basically shows how you can use patient tissue to show how if you have a mutation in an epigenetic modifier gene, that actually you may lose certain repressive pathways. We showed that PIDM5 interacts with major epigenetic modifier proteins and that these interactions are interrupted in the presence of a brittle cornea syndrome causing genes. And finally, I just wanted to highlight the candidate gene approach because we've all become very sophisticated now. And I really thank all the other speakers for their very sophisticated talks. But the candidate gene approach probably still has value when we're looking at very rare diseases. For example, at the moment, I have two individuals from two separate families with a very rare disease and pathological myopia. And again, it's a mutation in a transcription factor. And I'm thinking, oh, that's very interesting. We don't know much about transcription factors that make the eye grow. So I think that in very rare diseases, it's interesting to think about this candidate gene and how you could look. And also, next slide. It's important that through the candidate gene approach, we did show that there was significant enrichment for pathogenic alleles in the brittle cornea gene ZNF469 in keratoconus. And it was a very easy, simple study, which just involved a Holloda Sanger sequencing at the time, but it does remain one of my most highly cited papers. Not to say a lot, but, that was just through a candidate gene approach. So ZNF469 has been implicated and reproduced in all the genome-wide association studies since as being very important in corneal thinning. So next slide. I would like to thank you all very much for listening. I'm very happy to take any questions. And obviously I'd like to thank the patients for letting me show their pictures and donating their material. And thank you all for inviting me to speak today, although by mistake. And I hope I'll be invited to speak more than once. Thank you very much.